cells. What we were interested in our lab, one of the things, David mentioned that I spent a lot of time on hyperthalamus, which is true. But what we were interested is that how does this strange structure, this neuron, the nerve cell, which is the strangest cell in the universe, how does it come to pass? How is it formed? Like other cells, it has a cell body and a nucleus. It has receptive regions, which in the case of neurons have dendrites, so it receives the information. And it has a secretory process. Many cells have these processes. But what other cells don't have, and what is unique about the neuron, is it has this extremely long extension called the axon. And I think you all probably have heard about the axons, but I don't know if you realize that the cell itself is only, on average, about 1/1000th of an inch in diameter. Yeah? These axons are 1/10 to 100th that diameter, and they are perfect cylinders that run from this kind of amorphous cell all the way to tremendous distances. Just think of a whale's spinal cord and having to wave its tail. How long that axon is, and it's infinitesimally small, microscopically small. So the question is, how does this axon maintain its integrity through all the things it goes through? So when the whale waves its tail, the axon is being bent. When you run the Woods Hole Marathon, your axons from here to here, which is really long for this very thin thing, is being battered. So how does it maintain its integrity? How is it maintained? How does it develop? And so one of the things we knew, and of course Phil knew when he came, was that the inside of a cell has a skeleton too. We call it the cytoskeleton. Okay? And that what happens is between these little objects that you see, these filamentous objects, are filaments and tubules that are found all over the cell, but in the axon, they line up through the whole distance of the axon. Okay? How does this happen? What are the functions of these different tubules? And they're called microtubules, actin filaments, and neurofilaments, which was the obsession that we had in our lab. So Phil started in a project which was to consider in the frog. Now the frog is the animal we continue to use then. What were the functions of these neurofilaments? And nowadays, if you were to ask a question like that, what David has described, we have wonderful tools now in neuroscience and biology that you can do all sorts of stuff. So nowadays, you would simply remove these neurofilaments by genetic techniques, by molecular and genetic techniques, and you'd say, what happens? But in those days, we couldn't do anything like that. This is 25 years ago. So the first project that Phil did in our lab is based on this finding. Another developmental biologist who Phil knew very well, Marcus Jacobson, actually did this neat experiment. They injected, actually there's an element of that in David's talk as well, but they injected a dye. It's not important what kind of a dye, but it's a dye that can actually move from cell to cell that traces the lineage of the cells. In other words, every cell that's going to come from the two-cell stage in the animal will have this dye. Okay? If it's injected here, what did Jacobson find? He found that half the cells in the spinal cord, or in this part of the brain, all the parts of the brain, that one side came from this cell, and the other side came from this cell. So having that information in mind, a fellow in my lab named Ben Zaro and Phil did the following experiment. So this is an example of an experiment. Okay? And that is, they injected a dye, which happens to be a fluorescent dye, and antibodies, a whole variety of antibodies that could actually bind to and disrupt neurofilaments, the thing I was telling you. And the question was, when the animals grow up, what's going to happen? Are these neurofilaments just sawdust that fill this thing and have no interesting function? Or did they have other functions as well? And the experiment was, after injecting antibodies that should disrupt the filaments, you have the perfect control on the other side, where an antibody is injected as a control, which doesn't affect neurofilaments, and you see what happens on one side and the other. And to make a long story, because these are all long stories, but to make a long story short, what you get is something like this. So if you look at the result, and I'm only going to give you two things to look at. The control side of the spinal cord, in this case actually it's the cranial nerves, the 10th nerve, and you can see all these axons and stuff like that, and on the injected side. And you see it virtually all disappear. It didn't really disappear, this is an exaggeration of a result, but they become thinner and weaker and sometimes actually disappear. So it turned out, from this relatively simple first experiment, it turned out that we could make the following statement, that the inhibition, that is axons, were inhibited in development by the blocking of the neurofilament. And it says these data demonstrate that the usefulness of specific antibodies perturb neural development in intact frog embryos, and in addition suggest a role for neurofluorescent elongation. I should say this didn't answer the question, but it initiated a discussion, and this young man, who was young man at the time, Ben Zaro, went on to continue this work both in our lab and later in his own lab, and is now professor of biology at the SUNY Albany, and is continuing to work on this question and so on. So one of the functions that Phil had in the lab, just to make the bottom line of this, is as a mentor to various young people. He was a very inspirational mentor, just to see how Phil functioned, how he worked, such enthusiasm. I am actually saying something nice, but he actually played a very important role in that regard. So let me come to the second project. This was the first project he did. It was on his beloved frog. Okay? The second project was on this awful, fuzzy mouse. Okay? But the thing about Phil, when you talk about the curious mind, is that his enthusiasm in the laboratory is infectious. He's fearless. He's ready to work on anything, to try anything, and he did in this case. So here's an experiment, another experiment that he participated in a very important way. And it's about a cell type in the hypothalamus, which was brought up by David, the hypothalamus of a mouse, in this case, which has a cell called GnRH, which sends its axons to the base of the brain, where it secretes a product, which I'll tell you about in a second, into blood vessels at that base, which are a special private line of blood vessels that infiltrate the anterior pituitary. What this is, GnRH stands for gonadotropin-releasing hormone. It's a neuropeptide that causes the release and secretion and synthesis of the gonadotropins. That is, hormones released from the anterior pituitary, which go to the sex organs, and have the sex organs release estrogen and testosterone, depending on which section. Okay? And so this...
Hear that, Alex?
Sex organs? Why is he into it? So, actually, Alex, is very important. These cells, without these cells, there is no sex, there is no reproduction, there are no species. So these few cells in the brain are responsible for essentially the existence of all animals. So that student, this fellow who was working on this in our lab, had actually done her initial work in the University of Rochester. Her name was Susan Ray. And she had actually looked at what was the distribution of these cells. There were about 600 of them in the mouse, in the base of the brain, in the hypothalamus. And she found they were everywhere, which way. And the question was, at this point, where did they come from? The conventional wisdom, and by I meant where are they developing from? The conventional wisdom at the time was they were coming from the base of a ventricle, that is from cells that have stem cell potential at the base of the ventricle. This is still the current view in general about where brain cells come from, and that they spin out and then populate, they differentiate, and then they populate the brain. So this was the view. Susan Ray did experiments to try to determine if that was true. I mean, it was one of those things where you hypothesized what you thought was right, and it turned out to be completely wrong. What happened was, she met, as much as she tried, she could not find where those cells were coming from. At one point, she had the impression that they were coming from outside the brain and into the brain, and were coming from the nose, the development of the embryo's nose. To make a long story short here too, what Susan, and where Phil came in on this, is Susan did a series of experiments with Phil to provide evidence that these, is the alternative name to GnRH, luteinizing hormone, are derived from progenitor cells in the olfactory bulb. This was a revolutionary idea. It was the first time this was ever thought. And I have to say that there was one other laboratory during the same year published such a story, not as well done as ours, of course, but published such a story. And these two papers went on to essentially revolutionize the field. What in fact was happening was that cells in the nasal placate in the developing embryo were lining up and like little soldiers marching through the nose, okay, and moving into the brain parenchyma, the brain space, and populating it to the way that you saw in the first picture. How did they get there? And this is actually the actual data. I'm not going to actually talk about it. You can actually see them moving through the spot and moving into the brain. So how did they get there? Well, it turns out that the way they got there was walking along olfactory fibers. In other words, there are nerves that come from the nose that we smell with. These are the olfactory nerves. And that these little cells that are so important for our survival and species survival are schlepping along those olfactory fibers to get into the brain. So this, let's say, this was a remarkable finding. It sort of revolutionized the field. Susan became a leader in this field as a result, and a tenured person at NIH, and this continues to work on this problem. Okay. One of the byproducts of this finding, and as I say, Phil played an important role because he brought to us in this study a technique called thymidine labeling, in which you can determine what the birthday is. That is the last day that the cells divide. Without going into any details, those were critical for that part of our evidence. What was important about this work from terms of, somebody always asks, so what does it mean to humans? So what's important about this work for humans is it gave us insight into a human disease, and that was called Kalman syndrome. So what is Kalman syndrome? Kalman syndrome is a condition where people have no reproductive capacity, they don't develop primary or secondary sex characteristics, and the fact is they don't have these neurons in the brain. The neurons never got in the brain. But the second thing that these people have is anosmia. Which means they can't smell anything.
You can't smell
anything. How does that happen? And why can't they smell anything? Because they cannot put out those fibers, those nerve axons that go into the brain from the olfactory mucosa, from our nose, and therefore these cells have nothing to climb up.
So they can't have sex and they can't smell anything.
That's it, they got a problem.
Big
Big problem.
They can't smell anything and then they can't have sex.
So that was the second project that Phil actually, it made it actually the second project that you worked on, Phil coming some 25 years ago. So coming more to the present, this is my third story. And it involves, again, the issue of neurofilaments. But it derives from another passion. So we were talking about Phil's passion. And his passion is Woods Hole, right? There's no question that Phil has passions about Woods Hole. And the second passion he developed in Woods Hole in our laboratory was the squid. Okay? So I'm sorry Harish left because he's very important in this story. And also, Harish is also a source of very good squid for eating. And so it's really a beautiful animal, and to some extent it captures its beauty. It has these beautiful chromatophores which flash, it uses it in all sorts of behavioral ways and so on. But our interest was, again, to study the neurofilaments in this axon. The squid has the mother of all axons, the squid giant axon. And unlike what I told you before, which is true for most nerve cells, where the axon is very thin but cylindrical, in the case of the squid, the axon is enormous. It's seeable, you can see it with your eye. And it is because lots of cells have fused in the squid nervous system, have fused to make this one giant axon. Why does it have a giant axon? Does everybody know? Because it has to move fast and contract this mantle, and by jet propulsion, shoot itself fast. And a giant axon in the invertebrates is the solution they have for axons that have fast electrical conduction. Mammals and vertebrates in general have another trick, and I showed it actually on that first slide. I didn't comment on it, and that is a fatty deposit on the outside of the axon called myelin. And by doing that, we can make small axons that can move very fast. Invertebrates don't have that choice, so they make large axons. But squid is unique even in that regard, because there is no other animal, well actually there's one other animal, but it's not as good, a worm, that makes a really giant axon like this one. So what does it look like? Some of you probably heard this.
Can't eat a worm.
Pardon me?
Can't eat a worm.
Well, you can eat a worm if you're hungry enough. This worm is called Mexicula. It's a sea worm, and it has a giant axon as well, because it has the
It doesn't look edible.
And it also doesn't have the feature I'm about to show
Not
you.
as tasty and curry.
So what did you see as a giant axon? So here's probably a preparation dissected by Harish. He was a master, he became the master dissector in our laboratory. And there is from these cells that are found in this ganglion, which is expanded here, this very long axon. You know, it must be, in this case it must be about that long. But it's not the length that's important, because after all we have long axons too. It is the width. Because if you look carefully here, I don't know if you can see it, there's a translucent space, and that translucent space is the giant axon. There are actually several that come out of this, but we only use one. We use the biggest one. And that is an example of what it looks like when you first dissect it out cleanly and have some skills. And its unique characteristic and feature, and the reason we've come for 30 years to work on the squid giant axon,
In Woods Hole.
that's the other reason, because we can continue to be passionate about what's called and work in, is because it is the only cell in the universe in which you can squeeze it like toothpaste tube. You see the little here, there's a little, this is the nerve containing the axon, and here's the dissected axon, and by taking a roller and squeezing this, you can end up with axoplasm. This is the only place in the universe where you can actually isolate the cytoplasm of a cell. And this cytoplasm, and it's not just not only neurons, but certainly for neurons. And this cytoplasm, this axoplasm, we call it, has served us and other people as an enormous model to understand much about the biology of the squid giant fiber system. It is alive. It continues to be alive. It continues to transport proteins. It continues to metabolize. It continues to, what you were interested in, phosphorylate neurofilament protein. So what does that mean? Phosphorylation is where a phosphate group is put onto a protein, is attached to a protein, and changes the characteristics of the protein. This is called, the fancy word is post-translational processing, where a protein that is made can change its characteristics and properties by how much it gets phosphorylated. And the squid axon, Harish and I many years earlier found that the squid axon was a star at phosphorylating neurofilament protein. And then later we found in mammals, so it was a model for mammals. Later that mammals did the same thing. Okay, having done that, we could actually take that exoplasm and decide what is in it. And I remember doing an experiment with my daughter many years ago where we actually wrote a paper on the proteins of the squid axon exoplasm. But the most important thing for our studies was the fact that we could actually isolate, characterize, even clone by molecular biology eventually, 3 neurofilament proteins, which are not identical to what we find in mammals, but awful similar. So, along comes Phil into this company, and one day he walks into the laboratory, and he's holding what looks like a bunch of white grapes, okay? And he knows what I'm talking about. So it's a bunch of white grapes, and he goes to the, we had running seawater and all that, and he goes to the place, and he takes a Petri dish, and he puts these white grapes into it. And we say, what? Harish and I say, well, what's this? So he says, well, these, back to basics, these are the embryos of the squid. And we then looked at these white grapes. They're really not grapes. They were embryos. They were little eggs, basically, egg sacs. And looked under there, and what did we see? A miniature squid. It was amazing to see it. It actually had all those chromatophores. It was still flashing all those little chromatophores. It was teeny, little chromatophores. It even had the ability to do this escape response, you know, by squeezing its mantle and getting the water out. It must have had, we thought, a giant axon by that time. And Phil, being the, you know, quintessential developmental biologist, said, let's study it. And sure enough, not only did we end up studying it, but to this day, and I mean this summer, it's still being studied on our lab. Of course, the players have changed.
How big is it?
The little egg is probably a millimeter. You know, that embryo is probably a millimeter or so. So under the microscope, you can see one day. So now that you know what Phil does, one day in the summer, you can actually ask him to show you this under the microscope if you haven't seen it. So at the first paper that Phil did, and he led these studies in the summer lab, and continues to lead these studies on the developmental biology of the squid giant axon, and in particular, the neurofilaments in it. And in it, there are a number of players, including Bob Gould, who I thought might be here, and Harish, who was here, but Harish was, of course, became one of the leading people in our lab working on neurofilament phosphorylation. And this, as I say, continues. Let me just end with 2 more slides. And this is to say that he's one remarkable character, because at the age of 80 or so, he became an expert on the biochemistry of things. Because as I told you, these proteins are phosphorylated, and phosphorylation is a very important process in biology. And what Harish, especially, and Phil together with him, have began to work on those types of enzymes. This is a biochemical question that actually phosphorylate the neurofilaments. So this is another. It's beyond curiosity. It's actually curiosity, but with achievement. Let me actually show you the last slide, which I think is very, very important. And that is that Phil now, this is now 2006, when he wrote this article. You should be able to get it from him and read it. And I think it's readable. And that is, he has become the leading advocate, I think, in the world probably, for the idea that the squid giant axon system is a model for human disease, namely neurodegenerative diseases such as Alzheimer's. So this is a very interesting article which becomes the culmination of Phil's activity and continued activity in the lab. So as I say, I want to end with this slide, but I have only one more thing to say. And that is that I've watched Phil for 25 years, from the ages probably 65 or so-ish, to 90. And he hasn't changed. The thing is, he continues to be the same imaginative, creative, curious, as you pointed out, Gordon, passionate person as he was from the day he first walked in the lab. And the last thing I want to leave with you is that I really wonder, what will Phil be when he's 100? I suspect the same. Thank you, Phil.
I have to say something.
You have to correct me.
No, this is a response to all 4 talks. People recently come up to me and say, hey, you are 90 years old, and you think about death, and does it disturb you? And I say, yes, it does. I mean, of course you think about death at age 90. And I said, the only thing I think about is the fact that I'm going to miss the next 25 years in science. Because I think during that time, great things will happen in neurobiology. Great things will happen in nuclear reactors. Great things will happen in our understanding of water use. And great things will happen in development and
Yeah, and neuroscience.
and
Yeah,
neuroscience.
that's good.
And I'm going to miss it all.
No, they publish in heaven.
And you can still be the first
Don't
author.
forget to, don't
St.
forget
Peter's a senior
Well,
author.
don't forget to take your computer with you.
Okay.
Many years ago, Gordon, he doesn't remember properly, but Gordon and I were sitting somewhere at some party and we were pondering Phil's energy, enthusiasm, characteristics. And we both concluded that he was the beginning of a new species. Philip's the senses.
Well,
That was wonderful. Can I just, as the
Yes.
daughter of a scientist who cannot, to this day, ever explain quite what
What he does.
This was a brilliant, brilliant gift
That was
to
my
me,
jacket.
but also to his grandchildren. I mean,
Yeah, that's really
none of us quite understood this squid thing, and you know, I thought it was just merely good bait.
Except
You
it's
know, it tastes so
Good
great,
bait,
right?
I
Good
mean.
bait. But for you to weave it for us as a narrative that we can, you know, really absorb, it's how you did a beautiful gift. That's a gift. Thank you.
Are there any questions for Hal?
Oh,
Deborah, give one?
Well, I wondered if we were gonna have a test on this later.
no.
I have a question.
But you got the sauce. You can ask
No,
anything.
but it was beautiful actually, because I didn't understand what he was working
Oh,
on.
that's what I figured is the main
Yeah.
point I could
You
contribute.
don't talk about this stuff, right?
No, it's very difficult to talk to your family about it. A, you're not always that interested. And we may not explain it that well. And but it's a passion.
And it's hard to have
It's
a
really
slide
and this guy's got the passion. I mean, there's no question about it. And it's really being a scientist, not just in Woods Hole, but in general. And you got a feeling of it from David. It's just like a constant detective story. You're constantly involved in some mysteries. The thing that's amazing about Phil is that the rest of us are kind of down when it doesn't work out the way we like. Not Phil. He's ready to move on to the next one. So he has this enthusiastic characteristic, which is extraordinary.
That's great.
Yes, amazing.
Oh, so my question was, it seems inherently inefficient to have sexual characteristics, you know, sort of start off in the olfactory bulb and then make, it just seems like a, why would it, you know, or
Well, you know,
maybe
smell
it
and
seems,
sex
it
have been
seems
connected.
random.
Well, yeah, does it have to do with like pheromones?
Pheromones. Perfume.
Yeah.
So smell and sex are not completely out. They're not completely out to lunch. And you have that's also true in terms of the sense, not exactly smell, but certain kinds of type of input in yeast and sexual mating in yeast. They have also things that they secrete as a result of stimuli which involve mating factors and so on. So they're not unconnected, but I'll tell you, none of us had a clue that that was the way it was going to be. It was a complete, that's the thing about, for example, David's point about he looked to see and suddenly he found himself working on blood vessels. It wasn't his point, but it falls out of your pocket. And you sort of say, I never thought of this. Let's pursue it. Small-minded people, by the way, small-minded people think you shouldn't pursue it. You should stay in what you're supposed to do. One of the things that Phil and I share, I think, is screw them. We do what we like. And that comes out is usually pretty good. Because the real world is much better than our imagination in understanding how things are. So it's really, it's great. I can understand entirely Phil's desire always to go back into lab. Because it's a kind of an exploration, a life's exploration, which is very special.
No, the real world presents things that our imagination can't conceive of.
That's why I read nonfiction.
There are more things in the, how's that going?
Oh,
There are more things to heaven and earth ratio than a dreamt of
That's right,
in your
Shakespeare.
philosophy.
That
That's
in
correct.
your philosophy.
That's correct.
So, on that note, I'd actually like to turn the program over to Daniel, who is going to act as pop-up meister. These pop-ups were intended to be short statements, contributions